The DNA molecule that contains all the genes required to run and construct your body stands a massive 2 meters tall. That's almost as tall as Shaq. Yet all this DNA can be packed into a cell nucleus, just 1/50th the diameter of a grain of salt. We understand the importance of this DNA compaction to tumour development, stem cell and gene therapies, and viral infection. However, exactly how DNA is packed into the nucleus still eludes us. What we do know is that DNA is coiled around molecules called histones, but how these histones compress is not understood. Some researchers describe a hierarchical folding of histones into progressively larger structures. Other researchers think that histones are not organised at all, but rather just packed into the nucleus in small chains like beads on a string. How can we have such conflicting opinion over such an important question? How can structures that some researchers are observing not be found by others? What impact is this gap in our knowledge having on our ability to understand diseases like cancer or develop new treatments like gene therapy? As it so happens, this contradiction comes down to a difference in approach. The observation of large DNA structures that inform the hierarchical model came from methods that freeze the cell using salt solutions. The DNA is then cut up, read, and reassembled in 3D by a computer. These methods are indirect, and salt solutions have been observed to create large DNA structures that may not be apparent in living cells. When researchers use methods that avoid salt solutions and directly measure DNA structures in the cell, no large DNA structures or organisation can be found. Can it be the way that we are treating DNA that creates these larger structures? This presents us with a problem. We know that understanding DNA structure is really important for understanding how cells function and treating disease, but we need to develop more techniques capable of visualising DNA structure inside living cells. We developed a technique that allows us to visualise the density of DNA in 3D inside living cells. By using a fluorescent molecule that changes the way it emits light when bound to DNA at different densities, we were able to build 3D models of the cells we have analysed and identify regions of concentrated DNA from less compact regions. In the future, we hope to optimise this technique for rapid analysis and integrate it with technologies that sort cells that may be gathered from, say, a biopsy tumour. We may even be able to combine this technique with microscopy tools that let us determine DNA density in living skin, allowing diseases such as skin cancer to be better diagnosed without biopsy at all. Developments in live cell microscopy techniques will progress our understanding, diagnosis, and treatment of numerous cellular diseases. So rather than just take a snapshot of a cell at one point in time, we can take a shot of a living, active cell.